Before we start, I would like to give a few instructions. The Zoom audience, please post your questions in the Q&A box or raise your hands and we'll unmute you. The YouTube audience, please post your questions in the comment section. Now, I would like to invite Dr. Puran Singh Sajwali to introduce today's speaker, Professor Philip Rosenthal.
Thank you, Saujanya. Can you hear me?
Yeah, audible, good.
Great. Good evening, everyone, and welcome to CCMB Biologue. First of all, I would like to thank Dr. Philip Rosenthal for agreeing to give a talk in your Biologue. And then it's my pleasure and honor to introduce to you today's speaker, Dr. Philip Rosenthal. And I have been fortunate and honored to be associated with him as a postdoc and as his mentee for almost now 20 years. So currently, Dr. Rosenthal is a professor of medicine at University of California, San Francisco. Phil obtained his MD from the University of Michigan and then worked at the University of California, San Francisco as a clinical fellow and PDF in the departments of infectious diseases. And at the same place, he has been working on malaria, different aspects of malaria for almost 3.5 decades now, or maybe there are a few more years, which I haven't counted. So I know Phil for many aspects, not only for the malaria research he has carried out as a person, as a mentor. So that is something we can talk about later as well. But one of his best quality or his success in carrying out malaria research is that he has been successful in carrying out research on all the 3 aspects of a research problem, which includes basic research, translation of research finding into applications, and epidemiology of disease and treatments. And Phil's lab has made few key contributions into all these areas, which have led to products, which means anti-malaria treatments, on changing the regimen for anti-malaria treatment in Uganda, where he has been running a center for almost now 25 years, maybe longer. He has been authored in almost 500 publications, served as editor in several journals, and currently he's the editor-in-chief of American Journal of Tropical Medicine Hygiene. And because of his seminal contributions in anti-malarial chemotherapy and treatments, he's a member of the WHO Technical Expert Group on Malaria Chemotherapy. He chairs the Scientific Advisory Committee on Worldwide Anti-Malaria Networks. And with this, I invite Phil to deliver his talk on anti-malaria drug resistance in Africa. Phil, welcome to the CCMB Biologue.
Thank you, Paran. I should have said I hate long introductions, but now it's over. I'm glad it's over. It's a pleasure to speak to you today. Thank you very much for the invitation. This talk will mainly be about Africa. As you know, that's where the malaria problem is the greatest, and it's where we work, but it has a lot of implications for India also, as you'll hear as we go through. So my slides were working fine until now. Let me see. There we go. Okay. This is an old slide, but it's still pretty accurate. Of course, you all know the biggest problem in malaria in the world is in sub-Saharan Africa by far. That's where about 95% of cases occur, nearly all Plasmodium falciparum, which is the most dangerous malaria parasite. And that's what we'll be focusing on. We'll be focusing on Uganda right here. Can you see my pointer? Just give me a thumbs up or thumbs down, Paran.
Yes, we can see.
Okay, good. So our work today will be discussing Uganda, but you'll notice one of the other countries that still gets some yellows showing fairly high transmission intensity is India. Outside of Africa, the country with the most cases of malaria is India. Just a little bit of introduction, sort of good news and bad news. There's been great progress with malaria. A number of countries have just in the last few years been declared malaria-free. Remarkably, China is one of those countries. And many others are having relatively few cases now. They're moving towards malaria elimination. That's great news. But unfortunately, that's not the case for Africa, where the problem is the biggest. So we're doing better on the edges, but not so well in the heartland of malaria. This is a little bit old now, but from about 5 years ago, this is a list of the 10 highest burden countries in Africa and how they did between 2016 and 2017. All of them had increasing number of cases. We're going in the wrong direction. The only exception on this list was the one non-African country on the list of high burden countries, which is India. India is closer to this side. It is getting better. There is a hope for elimination by 2030. That's probably overly optimistic, but nonetheless, India is seeing progress. We're really struggling though in Africa, where the story is mostly we're going in the wrong direction recently. And you can get a really nice summary of malaria every year in the WHO World Malaria Report. It comes out every December, and the news from last December was not very good. Malaria cases, the numbers going up. Case incidents, actually, because since the population of the world is changing, you can ask whether the actual cases per 100 people is going up, and it was going down, now it's going up. Malaria deaths going up quite dramatically. We were getting down to saying about 400,000 deaths a year. Now we're back over 600,000. About 2/3 of those believed due to problems related to the pandemic, not COVID itself, but changes in our ability to manage malaria because of the pandemic. But it's not only COVID, and the mortality rate going up. So the general story is we were doing well until about 2015, and since then things have stalled and maybe even getting a little worse, which is very disappointing. So we have to look back at how we control malaria. A key is prompt, effective therapy. For almost 20 years now, our standard therapy has been ACTs, that is artemisinin-based combination therapy, a combination of a highly effective artemisinin drug and a second longer acting drug. We also use drugs to prevent malaria in travelers, intermittent preventive therapy, in particular in pregnancy. That's a standard practice in malaria endemic places in Africa, where women receive therapy every month. Mass drug administration, mostly still experimental. Other regimens related to IPT, such as seasonal malaria chemoprevention, are used in parts of Africa to prevent malaria in children. Control of mosquitoes is extremely important, but not the subject of this talk today, except we'll briefly touch on it later. And as most of you know, there is a new malaria vaccine that is exciting, but its efficacy is somewhat limited. And even though it's now approved, it's not going to be available in large enough quantities to use widely for a number of years. But it's not quite a game changer. There may be some other vaccines coming. But for now, we're still very dependent on drugs, which is the topic of this talk today. The key drugs, again, are the artemisinin-based combination therapies. Normally, when I make this list, I stop here. I don't include artesunate sulfadoxine pyrimethamine because it doesn't work that well, except, as many of you know, this is still a standard regimen in India. It works better in India than it does in Africa because we have less resistance to SP, but it still probably should not be the standard in India. I think many people agree with that it should be probably artemether lumefantrine, the most widely used ACT in the world. And we'll be talking about resistance to SP in a few minutes. We also use drugs for prevention, and there SP still has a major role in Africa. SP, old-fashioned sulfadoxine pyrimethamine, is still the standard for all pregnant women in malaria endemic countries in Africa. I just mentioned seasonal malaria chemoprevention. This is a new, somewhat new policy in areas with seasonal malaria of giving children SP plus amidiaquine, two old-fashioned drugs, not all year, but monthly during the transmission season. And VHA proparaquine is much studied, it's not really used yet, but much studied as it's another artemisinin-based combination that is studied for chemoprevention. How do we look for drug resistance? There are three main types of studies, all of which we're very involved in. Clinical trials are, of course, very important to see if drugs are working. We're involved in many, but I'm not talking about that today because we don't really have too much new to talk about. I will make the point that we really need new trials, especially in Africa, where we're worried about drug resistance. In vitro studies of drug sensitivity are standard. And when we study parasites directly taken out of a patient, we call that ex vivo rather than in vitro, but it's basically the same assays. And these are not so easy to do in rural Africa, but we have a major program that I'll talk to you about in a few minutes doing that. And then lastly, and easiest is looking for polymorphisms associated with resistance. When we know what to look for, and in many cases, we do know what to look for, we know what genomic markers to look for in P. falciparum, then it's pretty easy to get some DNA sequence and see what's going on. And we'll talk a lot about that. So what I'm going to start with today is talking to you about the last point, sequencing to understand drug resistance, with surveillance for drug resistance mutations across Uganda. We've been doing this for many years. I used to joke that it was boring. We were seeing some changes, but we weren't seeing really worrisome drug resistance problems. And that has really changed in the last few years, as you'll hear. Things have gotten maybe not so good for Africa, but very exciting for the researchers. So we've done surveillance originally going back even further, but by 2012, we had a standard program with 3 sites, which were here, here, and here. Tororo, Jinja, and another site here. Moved to 10 sites in 2016, then 16 sites, which are shown here, led in recent years by Victor Asua, who's now a PhD student with our program. And Victor has collected in recent years 50, and then we moved it to 100 samples once a year. Very simple. We just go to sites, collect samples from people with malaria. Nothing complicated about the clinical study. And then we sequence genes of interest. We've used a number of different technologies for sequencing. As I was mentioning before the talk started, most recently, mostly molecular inversion probe, which is a related to amplicon deep sequencing approach that allows us to sequence multiple, it's not whole genome sequencing, but allows us to sequence something like 50 genes at one time in a multiplex format, and it's working quite well. And I just mentioned that we know what to look for. This is what we look for primarily. And then on a secondary level, we look at many other genes. We know that 2 transport proteins, PFCRT, PFMDR1, are mutations in those proteins are closely associated with resistance to aminoquinolines, such as chloroquine and amidiaquine, and they have an impact on other drugs. With antifolates, sulfadoxine pyrimethamine, we understand this very well. Mutations in the 2 target enzymes, DHFR, DHPS, are associated with resistance. And with artemisinins, very important work in Southeast Asia, something like 7 or 8 years ago, identified the key mutations in the PFK13 gene that are associated with resistance. And we'll talk a lot more about all of these. First of all, the transporters, again, this is old stuff. We've known about this for a long time. Resistance to chloroquine is primarily mediated by a single mutation, the 76T mutation in the PFCRT gene. And secondarily, this mutation in particular, the 86Y mutation in PFMDR1, plays a role. Important for chloroquine, which we don't use much anymore, but also, though chloroquine, of course, is still used for vivax malaria, but also for amidiaquine, which is a component of the second most commonly used ACT in the world, artemisinin amidiaquine. Piperacin, the other important aminoquinoline, is affected, but it's more complex. It's somewhat different mutations impact on piperacin. Interestingly, 2 other drugs, but most importantly, lumefantrine, the partner drug for the most widely used ACT, artemether-lumefantrine, is affected in an opposite manner, such that the mutations that lead to resistance to chloroquine lead to increased sensitivity to lumefantrine. And so that helps us understand what's going on with lumefantrine. There is no high-level lumefantrine resistance in the world. That's the good news. But these mutations have some effect on lumefantrine. So we've been studying these for years. In the old days, back 15, 20 years ago, all parasites were resistant. When we first started working in Uganda, parasites were 100% resistant to chloroquine. But we've watched a dramatic change, especially over the decade from 2010 to 2020. You see PFMDR, and in this case, we looked at 3 different sites and 3 different years. And wild type parasites are in red. You see the prevalence of wild type dramatically increasing between 2012 and 2015 at all 3 sites for 86Y. And for 76T, the primary mediator resistance, the process happened later. We were just starting to see wild type in 2012, but the same pattern, increasing resistance. I'm not going to talk about these other 2, which are relatively unimportant, just to save time. Moving a few years later, sorry, I'm showing things a little different way. Each time we publish, we do it a little differently, unfortunately. Here's maps for 2018, 2019. You see mostly light color, meaning no longer seeing high prevalence of mutations to 76. That's up here. Still a few sites in 2018, but not so much 2019, that we still see 25, 40% resistance prevalence, but it's going down. And the other one, 86Y, is pretty much 100% wild type now in Uganda. So not much to say about chloroquine resistance. It's mostly gone away. You can use chloroquine to treat malaria in Africa, and it would usually work. I wouldn't recommend it, but it would usually work. Secondly, antifolate mutations. As I said, we understand them really well. These are inhibitors. Sulfadoxine inhibits DHPS, pyrimethamine inhibits DHFR, and we know from work in the laboratory back in the 80s and 90s and in the field long ago, we know that there's a series of mutations that are very common in Africa. 3 mutations in DHFR, 2 mutations in DHPS. In where we work in Uganda, almost all parasites have these 5 mutations, as I'll show you in a second. Curiously, in West Africa, one of these mutations is absent. That's 540E. These are haplotypes that don't include 540E. These are haplotypes that have the 5 mutations I just mentioned. So in West Africa, there's less resistance to SP. These other mutations don't lead to much in terms of clinically important resistance. And SMC with SP plus amidine, seasonal malaria chemoprevention, is widely used now in areas of seasonal malaria in West Africa. But if they get 540E, it probably won't work as well. And even more concerning are even more mutations. The 164L mutation and the 58 in DHFR, 581G in DHPS. These would be the 6th and 7th mutations, which lead to very high level resistance, and probably SP having no antimalarial activity. So as we were studying those other mutations, we're also studying SP. This is looking again at in 2016, 2017, this is when we were at 10 sites around Uganda that we studied at 3 different times. So 1, 2, 3 here are 3 different times that we studied these. 5 mutations are almost 100% mutant. We knew that, nothing new. But what we are seeing are the beginnings of 164L and even more so 581G popping up. And if you knew malaria, Uganda geography, you know these are sites in the Southwest, showing that now from a couple of years later, 2018, 2019 samples, we're seeing the 164L in 2018, 2019, and the 581G mutations, primarily in southwestern Uganda. Interestingly, right up against the border with DRC, Democratic Republic of Congo. And here's a map of DRC. This is done by Jeff Bailey, John Giuliano's group. And they showed, using samples from all over the country, that the 581G mutation was at high prevalence right next to the Uganda border. There's some big mountains in between Uganda and DRC here, but there are ways around the mountains, and clearly people are bringing the parasites to both sides of the border. And most recently, actually, this map does not include 2021 because we haven't gotten around to updating the map yet. We've been spending our energy very recently on Artemisinin resistance, not SP resistance. So I'll update the map shortly. But basically, sporadic resistance around the country, but especially in the Southwest, right around here is where we're seeing problems with both 164L, 581G, but it is spreading through most of the country. So it's making it look like SP will probably have no antimalarial activity. But now we move to where the excitement has really been, which is Artemisinin resistance. And I'm trying to teach myself to use the term Artemisinin partial resistance. There was a lot of energy spent trying to figure out the best term to use for artemisinin. A lot of people don't like the term Artemisinin resistance because it's not full-blown resistance. And we've ended up with the term Artemisinin partial resistance, which I don't like very much, but I'm trying to use it because there's a WHO consensus panel that decided it's what we should use for this term. This was discovered a little over 10 years ago, when it was seen that it took longer for people to get rid of their parasites. Typically, these two curves show studies here in Thailand, in northern Thailand. And when you treated people, their parasite load went down over a couple of days. And this is with two different courses of artesunate, one of the artemisinins. That's normal. And what they started seeing is people taking longer to get better. They still got rid of their parasites, but it took a few extra days, was delayed clearance. That was first seen in Western Cambodia and clearly was very important. It's not full-blown resistance. The parasites still respond, but they respond very slowly. That's what I'm going to be calling artemisinin partial resistance. It's an enormous problem in Southeast Asia, and it's been around for quite a while now. So we know it very well. This is an area with major problems with drug resistance, but not really major problems with malaria. There isn't that much malaria in Southeast Asia, so it's not a catastrophe, but it's very worrisome because if this spreads to other parts of the world, such as Africa or India, it could have very major implications. This is an old map, but it shows the places that have red in their pie charts have the mutations and also clinical evidence of delayed clearance, focused primarily on Cambodia, but really throughout Southeast Asia, but not India, not Bangladesh, only the parts of Myanmar on the near India that have this problem, and certainly not in Africa. With this problem, we see decreased efficacy of artemisinins, and if you lose also the partner drug, then you get true resistance to the artemisinin-based combinations, as has happened in Cambodia with some drugs. Typically, clinically, you see delayed parasite clearance. A standard IC50 assay, the way we normally measure drug susceptibility, doesn't work well to recognize this partial resistance phenotype. And so another assay was developed by a French group in Cambodia called the Ring Survival Assay. I won't bother to go into the details unless anybody wants to hear it, but it's a different type of assay that recognizes the abnormality in rings that is associated with this phenotype. And that same group, that French group in Cambodia, also recognized the really key finding, which was, sorry, with the Zoom, my parts of my screen get blocked. That was Aria's paper from 2014, which identified mutations in the K13 gene as the key to artemisinin resistance. A little bit of more basic science. I know a lot of you are basic scientists. Really interesting stories about how K13 mutations mediate artemisinin partial resistance. We don't fully understand this. We're still figuring things out. When K13 was discovered as the mediator, nobody had any idea what it did, but it's a Kelch protein. That's where the K comes from, a family of proteins, in many systems. And it has this big propeller domain. That's where the mutations are. But what we've learned is certain mutations in this protein is involved in the process of hemoglobin uptake by the parasite. The parasite takes up hemoglobin from the red cell and breaks down hemoglobin in the food vacuole to provide amino acids. And it's seen that these K13 mutations decrease hemoglobin uptake, especially in young ring stage parasites, the early stages of the parasite after they invade the red blood cell. And that leads to decreased hemoglobin available in the food vacuole, and it's the breakdown of hemoglobin that's required for activation of artemisinin. So when you slow down this process, you get parasites that are relatively resistant to artemisinin. That's what we understand about it so far. There'll be more work on that. This is really nice work from Toby Spielman's lab that was just published recently. But I want to mention that the basis for understanding this, a key part of that, is understanding what goes on in this food vacuole. We know that, as I just said, yeah, I'm having trouble seeing this. Those of you who've given Zoom talks, you always get the pictures of all the audience get in the way of your slide. Hemoglobin hydrolysis is required for artemisinin action, as I just said. Among the key enzymes breaking down hemoglobin are phosphamine 2 and phosphamine 3 cysteine proteases in the food vacuole, and loss of phosphamine 2 activity leads to artemisinin resistance. This last point was shown by Clonis in Leon Tilley's lab about 10 years ago, using both parasites had phthalate 2 knocked out and also inhibitors, but all of the work that led to our understanding is due to the work of Paran Sijwali, who for multiple years in San Francisco did beautiful work characterizing phthalate. Not alone, there was a great group. Actually, it turned out most of the folks working on this were from India. It wasn't really chosen that way, but that's how it happened. But Paran really led the effort and did. The really important point here is that basic science done between about 2000 and 2010 led to really important understanding many years later that are really of practical relevance to addressing drug resistance today. That's as basic as this talk gets. Now we'll get a little more to translational work. This is what we know about Artemisinin partial resistance. As I said, it is delayed parasite clearance in vivo in patients. People typically still have malaria parasites on day 3. Otherwise, without resistance, they're virtually always cleared by day 3. You can calculate that as a half-life. A half-life more than 5.5 hours is considered partial resistance. And we have this nice in vitro assay, the ring survival assay. Parasites surviving better than controls at 72 hours after a pulse of artemisinins have the in vitro evidence of delayed clearance. And importantly, as I told you, mutations of the K13 gene are associated with resistance, but it's not that simple. There are many mutations in the K13 gene, actually over 100 different mutations. We generally only see, I should say, in the propeller domain of the K13 gene. We're not really talking about the entire gene. And many of these mutations occur now and then, but don't seem to have any importance. There are about 20 of them that are considered candidate or validated mutations. This is a little bit out of date, but there are about 20 that are validated. And we're going to be focusing now on, well, first of all, I'll say that 580Y has become the most common. Every one of these mutations was first discovered in Southeast Asia and shown to be associated with delayed clearance. That's why it's on the list for validated or candidate. 580Y has become the most common in Cambodia. It's replaced others due to fitness advantages, we think. And then the others that are circled here are mutations that have popped up recently in Africa. But let's digress a moment and talk about India. There were really interesting studies published, it's about 7-8 years ago now, from West Bengal, with 2 studies that were really sort of the same study over 2 different time frames, published in these first 2 papers. And they showed very worrisome delayed clearance from Eastern India with patients with persistent parasitemia on day 3, abnormal ring survival assays, and K13 mutations. One of them already on my list, 539, is here. Well, actually, it's not on the list, but one that is known to be associated now, and the other, a novel mutation discovered in India. And then a second study was really just a bigger version of the same study, showed the same kind of stuff. The same group also showed some delayed clearance even without K13 mutations, another interesting result, and we'll get back to that in Africa later. These are fascinating results that are very concerning, but it seems, it's not really published, it seems that other groups have had trouble replicating this data. So it's really, most people do not think Artemisinin resistance has a foothold in India, even though these papers imply it does. We'll see how this sorts out over coming years. But to my knowledge, there are no other reports that support these findings. Not sure what that means. But looking around the world, we have somewhat similar stories elsewhere. A few other places where some mutations in K13 are seen everywhere, just at very low prevalence, maybe 1 or 2%, don't mean too much. But when you see the key mutations like 580Y, that gets you concerned. This has been seen in Guyana in South America, a number of parasites with the 580Y mutation, but there isn't that much malaria there, and this has sort of gone away, it seems. In the most recent studies, from what I've heard, it hasn't really become sustained. And in New Guinea, Papua New Guinea, there have been a few parasites also with 580Y discovered, but again, haven't heard much more about it. So it seems that there are these tantalizing reports, but nothing that I know of suggesting sustained prevalence of Artemisinin resistance mutations outside of Southeast Asia. until recently. And now we'll talk about Africa. Older study, our study, of course, we were looking for Artemisinin resistance through the years in Uganda, and we really didn't see anything. And we have papers that very strongly say no resistance, published not too many years ago. Now they're out of date. We saw low prevalence of K13 mutations, and we did not see the most worrisome mutations. And we did ring survival assays, and they were fine. So as of 2016, 2017, we weren't seeing any problem. Then things changed. And as we ramped up our surveillance, looking at 10 sites around the country, we started to see 2 mutations. I should say, let me back up and say, first, we knew about a story from Rwanda. Rwanda is right down here, right next to Uganda, small country. And they started seeing the 561H mutation, prevalence up to 20% in multiple studies. So it's not just a brief transient event. And they did see clinical delayed clearance associated with this mutation, also abnormal ring survival. So real, what we now call artemisinin partial resistance. Meanwhile, in Uganda, we were seeing these 2 mutations, not the same mutation, but rather 469Y and 675V here in 2018 and 2019, increasing prevalence only in one part of the country, up in the north and northeast. Now we've looked very carefully. We've gone back to look at some older samples and gone forward up to 2021. And this is what's going on. It's a lot. 469Y and 675V, both were just starting to show themselves up here, way up in the north. This is Lamwo province up near Sudan. And then starting to increase, but pretty much all in the north, all through the years. Still up to very high prevalence in the north, now with a little bit of spread to other parts of the country. And yet there are 2 more mutations. There's another mutation at this same codon, but a different mutation, 469F, that we're seeing in a different part of the country. It was sporadic, but now really becoming deep-seated in the southwest. And 561H, the mutation previously discovered in Rwanda, we didn't see it at all until just 2021, and now it's pretty high prevalence here. And in fact, this site here, Rukiga province, has about over 50% prevalence of one of these 2 mutations. And Lamwo province up in the north has over 50% prevalence of either of these 2 mutations. So these are high prevalence mutations now in 2 different parts of the country. This is going back, this is what we know from other groups. In Rwanda, as I said, the 561H mutation seen at up to 20% of isolates at 2 different sites. Patients who had persistent parasites on day 3, about 10% of patients had persistent parasites on day 3 in a clinical study, and most of those patients had this mutation. So it is associated with delayed clearance. Though it is important to note that when they were treated with artemether plus lumefantrine, the treatment efficacy was okay. We do not yet know that this has a clinical consequence, but it's very concerning. And there was an important study published in the New England Journal by another group who works in Gulu, not too far away from where our sites are in northern Uganda. I'll show you that Gulu is about right here, so it's near these sites. They did a clinical study and showed that about 5% of patients treated had delayed clearance, and nearly 13 of 14 of those who had delayed clearance had one of these 2 mutations that we had seen a few years earlier. In the Horn of Africa, nothing's published yet, but there will soon be a paper published, something that's already been presented at meetings, so I don't mind talking about it, that there's yet another mutation, 622I, seen in Eritrea and parts of Ethiopia that has a similar story, also associated with delayed clearance. So we've looked further at the parasites from Uganda very recently, 2 different types of dendrogram analysis. This led by Melissa Conrad in our group, looked at microsatellites flanking the K13 gene in parasites collected over the last few years. And it looks, the important question is, are these parasites imported from Asia? Did somebody fly from Cambodia, bring them to Uganda? And it looks like the answer is no. It looks like these are independent emergences. These parasites 469Y parasites are closely related to each other, different than the wild type parasites. 675V, there might be two emergences. These are all northern Uganda parasites. These are from western Uganda. Maybe there are two emergences, maybe it's one. And then the 469F seen in southwestern Uganda clearly are more closely related to each other. But comparing with Asian parasites, we only could find Asian parasites with one of these mutations, the 675V, but the Asian 675V parasites are this is now looking at polymorphisms across the genome using a molecular inversion probes. And the African parasites are all very distantly related to the Southeast Asian. So all of this supports local emergence of African parasites with partial resistance. Why did artemisinin partial resistance emerge in northern Uganda? I have a kind of complicated story to tell you, and I realize I'm a little short on time, but we think the key is recent interventions that ended up causing problems. We have the hypothesis that resistance selection was facilitated by stopping indoor residual spraying, the really effective mosquito control measure, by stopping IRS after a long period of sustained and effective malaria control, which had allowed malaria to come back with malaria epidemics in a relatively non-immune population. So these districts in red up here all used IRS from 2010 to 2014. Malaria came way down, it worked very well, but then they stopped. The country cannot afford to do spraying all over the country. So they thought they had solved the problem. They moved to other districts. They started spraying down here. These places had a dramatic resurgence of malaria, but they were a relatively non-immune population because they hadn't had malaria for 5 years. And we think that is the perfect storm for selecting for resistance. And I'll show you that further on this. We just spent about a month working on the best way of telling this story. We just submitted this for publication on Sunday. And this is what we came up with. It's complicated, but I hope you guys can follow it. These are 3 districts up in the north, up here, where they got spraying with IRS every 6 months for about 5 years until 2014. Then they stopped. Malaria, and the blue line shows you one way of measuring malaria incidence. Malaria came roaring back. Then they sprayed one more time a few years later. Malaria went down, and then it came roaring back again. And you see that here, and here, and here. And the histograms show the prevalence of the 469 and 675V mutations in northern Uganda. You see, with malaria coming up, a relatively non-immune population because of the prior spraying, we started seeing prevalence of these mutations. 2 other sites where they started spraying in 2015, malaria went down, stayed very low, no mutations. Same thing here. Though interestingly, very recently, they switched to a different IRS insecticide that pretty clearly doesn't work well. It's very unfortunate. Malaria has skyrocketed, and maybe that's the reason we're starting to see mutations. I don't mean to imply it's so simple, but that's what we're seeing. These are 3 sites that have never received IRS, parts of the country that have not received it, and malaria is up and down, but in general, minimal selection of resistance. Looking in more detail at one site, this shows 3 metrics. The blue, again, is incidence of malaria. The green is another measure of incidence, total parasite rate, which show, I won't go into the details because we're running a little late, but 2 measures of incidence show these peaks, these epidemics of malaria. The red shows median age at malaria. It's a indirect measure of immunity. When a population has had a lot of malaria, they're very immune, and it's only young children that get malaria. But as the population is less immune, they haven't had much malaria, we see malaria in all ages. And so here's a population that was relatively non-immune after years of spraying, and non-immune population suddenly getting an epidemic of malaria. That was the recipe for selection of resistance. Looking at another site, Tororo, where they got lots of spraying between 2014 and 2020, malaria went down, the population became less immune, but there was very little malaria, so there was little opportunity for selection. Now, just very recently, they are using the wrong insecticide. Malaria has come back, and maybe that's evidence for what's going on. We really are interested in the 2022 data, and this preparation for sequencing is going on right now in Uganda. So we may have data within a few weeks to see what's going on. So basically, there have been complex changes, decreasing resistance to aminoquinolines, increasing resistance to SP, and selection of 4 different validated markers of artemisinin partial resistance in Uganda. I haven't had time to talk about it, but some key markers of resistance to partner drugs, which have been seen in Asia, have not been seen so far in Africa, which is good news. But we really don't know much about ACT efficacy. We know in studies a few years ago, artemether lumefantrine was still working well, but we really need new drug efficacy studies. These have been delayed a bit, in part because of COVID, but they should be coming later this year, early next year. And of course, continued surveillance is greatly needed. So you'd expect me to say that because that's what we do. So we certainly want people to support what we're doing. Now I want to talk about the ex vivo side. How do we define resistance when we culture parasites? Typically, a simple IC50 assay, 50% inhibitory concentration. You develop a dose response curve. But for artemisinins, as I mentioned, we have to look at the ring survival assay. We've been doing this in Uganda since about 2010. We routinely culture parasites and keep track. Each of these points is a single parasite isolate collected over a few years. And we look at IC50s. For most drugs, there's always a bit of a range, but we're generally seeing highly susceptible parasites to, these are most standard animalarials, piperacin, for example. But with chloroquine, we see sensitive parasites down here and still a few resistant parasites up here. We have seen changes through the years, most notably the stuff I've already talked about, the loss of chloroquine resistance. Our oldest studies from 10 years ago, chloroquine was we saw mostly resistance to chloroquine, IC50s, hundreds of nanomolar, now 20 nanomolar, amidiaquine, related drug, going from the resistant range to the sensitive range. You see that here. Chloroquine, amidiaquine, and to a lesser extent, piperacin, all getting better. I mentioned earlier that lumefantrine is affected in an opposite manner by mutations. So it's not surprising that as we've seen less chloroquine resistance, lumefantrine susceptibility has gotten a little bit worse. This is statistically significant, but clinically probably irrelevant. These are still highly sensitive parasites killed by low nanomolar lumefantrine. So not really clinically relevant, but we are seeing some changes in the wrong direction for lumefantrine. I will mention we also do this kind of study now in Burkina Faso in West Africa, where I was a couple of weeks ago. Not reporting any data from there, but we're seeing similar results there with parasites sensitive to the traditional antimalarials. And showing you the explanation for the changes between 2010, the explanations for this are shown on the next slide, which are what I've already shown you, dramatic increases in the wild type for K76T and the other key mutations, especially N86Y. We already talked about that. We still see roughly 100 nanomolar is the cutoff for resistance. We still see a few chloroquine-resistant parasites in Toro, Uganda, but not too many. But what we're really interested in now is not Tororo. We're interested up here where the action is, where we see the mutations, 469Y, 675B mutations. So we started a kind of crazy program where guys from our lab in Tororo, I should mention, this is where our main lab is. Guys from here drove up to Patango Health Center. What is that? 378 kilometers, twice a week, spent the night, the next morning collected isolates, and malaria is so common that you get new cases every day. And later in the day, we drive back, bring the isolates to our lab where we would study them. We did that twice a week for a few months last year and a few months this year. It's not sustainable. It's very expensive and takes a lot of time, but it allowed us to get some really valuable samples. The good news is we are now developing a lab up here at Kalango Hospital, just a few kilometers away from this health center. This shows our visit to the lab recently. Nice lab at this very nice hospital, but kind of limited in its infrastructure. So we were lucky to get a small grant from the Gates Foundation to allow us to build up this lab so that within a month or two, we'll be able to culture parasites up here, right near where we can collect them. What have we found? We found that, not surprisingly, the parasites, so then we compared parasites just from collected for a couple of months last year. We now have data from this year. These are last year's data. We collected parasites for a couple of months from that site in northern Uganda or from our usual place in eastern Uganda. Not surprisingly, these mutations were much more common in northern Uganda, though we are starting to see them now in the east also. We expected the ring survival results to be much more abnormal in northern Uganda, and we were surprised to see that they really weren't. We're seeing abnormal ring survival in both places. We did see a modest association between the 469Y mutation and prolonged ring survival, but it was a pretty modest association. And really, I think the most interesting thing coming out of our results is not high ring survival associated with these mutations. We already knew about that. It's some parasites with high ring survival not explained by K13. We have made some inroads into this, and this is primarily due to the work of Melissa Conrad in our group, showing that mutations in other genes are also involved. And remember, we're doing molecular inversion probes, so we're actually sequencing about 50 genes in the parasites of interest. And looking at two others, we see some interesting results. Coronin is a gene for which mutations were selected by artemisinin in the laboratory by Diane Wirth's group at Harvard. Haven't been seen in the field, but we just looked for any mutation in coronin. It is a somewhat polymorphic gene. And we found that having any mutation in this gene was associated with artemisinin delayed clearance. And if you had a mutation in this gene plus the 469Y mutation, there was the most strong association with delayed clearance in the laboratory. And this is falcipain 2, Poran Sejwali's gene or protein associated with delayed clearance, and in particular associated when we looked at either it or the K13 mutation. And in fact, if parasites did not have either the this is any mutation in falcipain 2 or any mutation or the 469 mutation, if they didn't have either mutation, they always had low ring survival. So this is probably overly simplistic, but it looks like every parasite that had delayed clearance either had the 469Y mutation or falcipain 2 mutation. So falcipain 2 does appear to have a secondary role in mediating Artemisinin resistance. We also, very concerningly, saw problems with lumefantrine. We didn't expect this. We were concerned about artemisinin, but interestingly, we also did routine IC50 assays on these parasites from northern Uganda, and they were less susceptible than the parasites from eastern Uganda to lumefantrine. This is showing this in graphically. Parasites from northern Uganda had a higher IC50, they were less susceptible, and all of the extreme outliers were from northern Uganda. 14 of 49 parasites from northern Uganda had high IC50s, and none from eastern Uganda. We're fascinated by this, very interested in studying these parasites, and yet, unfortunately, it's been difficult when we grow these parasites in the lab and they have relatively high IC50s. After we culture them for a while, we tend to lose the dramatic phenotype. This is after culture. This is after we culture adapted them, froze them, thawed them, restudied them. Our interesting phenotypes seem to go away. We suspect that these are all mixed isolates. Parasites are typically combinations of different strains in patients in Africa, and the more resistant parasites are the ones that don't do as well in culture. So we still don't have a good handle on what's mediating these elevated IC50s. This is not a real high number, but it's a little concerning. Some of these parasites, we might see problems with Artemis or lumefantrine for treatment. Again, why we really need to do therapeutic efficacy studies. This is just some newer data showing the same thing. We don't have a good explanation for this, but Melissa led analytical studies to try to identify among the many genes we sequenced, mutations associated with lumefantrine, decreased susceptibility. And all we came up with, there are a bunch of genes with modest associations, not sure if this means anything, but the really dramatic associations were K13. We don't think that K13 mutations directly mediate changes in lumefantrine, though it's possible. though we don't understand biologically why that would be. But nonetheless, there's an association, very strong association between the two K13 mutations we know about in northern Uganda and decreased susceptibility to limb infection. Just want to say we need new drugs also for malaria. Medicines for Malaria Venture has a major program for new drugs. Unfortunately, it's been very difficult to develop new drugs for malaria. We always seem to be about 5 years away from the new drug, and that's probably where we are now. It may be in about 4 or 5 years, we may start to see some new combinations. I don't have time to talk about it, but I'll just mention, we are also studying many new compounds with MMV. These are not labeled on purpose because some of these are secret, but these are results for many of the new compounds in the MMV pipeline against parasites in Uganda. And we're now also doing this in Burkina Faso. And Oriana Kreutzfeld is doing sequencing of the predicted targets of various proteins and looking for associations between polymorphisms in African parasites and drug susceptibility. We tend to not see high-level resistance to drugs under development, of course. They all work well, but we are seeing some associations. I don't have time to talk about it. This is already published with PFATP4 inhibitors, some association between certain mutations in PFATP4 and susceptibility to some inhibitors under development. Wish I had more time to talk about that, but the hour is almost over. I want to leave some time for questions. So I thank a huge number of people involved in these studies, and I'll be happy to answer your questions.
Thank you, Phil. Thanks a lot. Very exciting talk. Lots of new information to me as well, and I hope there's a lot of information for other participants as well. So now we are open for questions. And so if you have questions, please raise your hand. So there's a question from Manas. And Phil, his question is, what are all the pathways which K13 mutations affect? I think that's part of his question 1.
Yeah, well, again, our understanding of everything about K13 is limited. We don't really know its function, but it's been shown. If you want to look at one really good paper, look at the paper from Toby Spielman's group published in Science, I think it was 2020. And K13 is localized to the cytostome, which is the organelle that takes up red cell cytoplasm and pulls it into the parasite and brings it to the food vacuole. And it's involved there, and it appears to, with mutations, it doesn't work as well. It leads to decreased uptake of hemoglobin. Parasites survive, but they are less susceptible to artemisinins early in their life cycle, and that's the key. That's about all we know, to my knowledge, anyway. But clearly, the full role of this protein is not yet understood.
And his second question is, do all type of K13 mutations directly give or confer resistance to artemisinin?
Yeah, that's a good question. So the funny situation is, just talking about the propeller domain. Outside the propeller domain, there are mutations. They don't seem to be important in terms of drug resistance. So leave them out of the conversation. But considering the propeller domain, which is a few hundred amino acids, it's very common to see mutations, not very common, fairly common to see mutations. If you did an analysis of Indian parasites, you'd probably see 3%, something like that, would have mutations. But they're all over the place, many different single mutations. It's usually only one mutation. We don't really understand why. Resistance to many other drugs is caused by sets of mutations, but here it's usually just one mutation. But most of the mutations that have been discovered don't seem to have any consequence. And it's a small subset of about 20 of them that seem to be capable of mediating resistance. Though, our understanding of this is changing over time. We're gradually identifying additional mutations that might mediate resistance. But right now, there are about 20 that worry us and multiple others that don't appear to play a role. And most likely, obviously, this is due to structural features engendered by specific mutations in the protein. And, we need to understand that better.
Great. So the next question is from Dr. Maluka Maruti from, I think, Central University of Haryana, if I'm correct. So his question is, which is the best method to assess drug sensitivity? Is it IC50 or RSA?
For most drugs, IC50 is just fine. It's worked for many decades, and it's the appropriate way to look at drug susceptibility. For artemisinins, we know that the parasites that have delayed clearance in patients, if you did a routine IC50, there would be no difference. That assay just doesn't work. And we know that's why. That's why they call it partial resistance. These are parasites that behave normally if you incubate them with drug for the full life cycle. So the RSA, you only incubate for 6 hours with a high concentration of drug, then remove it, and then look at the parasites 72 hours later. That's the assay required for artemisinins. Also, for one of the partner drugs, piperaquine, sensitive parasites act normal in an IC50 assay, but when they're resistant, they act funny in an IC50 assay. You can't get good values. And there's a new assay somewhat similar to the ring survival assay that's specifically designed for piperaquine. So for certain drugs, we need other assays. For most drugs, IC50 is a good solid assay that I would recommend.
The next question is from Preetham. His question is, how does artemisinin application induces dormancy?
I didn't understand that, and I think
so. His question is that the artemisinin parasites are not completely resistant, so they probably undergo dormancy during artemisinin treatment period. So how is that dormancy induced? If I understood his question correct.
Yeah, the dormancy, that that's the terminology we were using for a number of years in trying to understand artemisinin effects. I think the easiest way to think of it is not so much, I don't want to get into the specifics. This isn't really my area of expertise, but I think the best way to think of this is not so much dormancy as just that with our newer understanding of K13, it's that the mutations lead to ring stage parasites being relatively resistant. So that dormancy is parasites in the ring stage that are relatively resistant to artemisinins. And so with treatment, with artemisinins, which only have a short half-life, only last a few hours, parasites are hit for a relatively short time, and they tend to evade the drug. So exactly how that happens, the best we understand is what I mentioned about K13. I emphasize that K13 is not the whole story, as I hope you noticed in my data. And so there are probably mutations in other proteins that help lead to this phenotype, which we could call dormancy in rings, we could call decreased susceptibility in rings, that allows people to get around artemisinins. What we don't see is full-blown resistance to artemisinins. That would be really, really worrisome, but that we do not see.
So the next question is it's a follow-up question from Manas again. So his question is, if K13 mutations decrease hemoglobin uptake, does it affect amino acid requirement of the parasite?
Well, that's a good question. There's decreased uptake. You'd think the parasite might starve from lack of amino acids, but as you know very well, Paran, there isn't really that much hemoglobin breakdown in rings. And in fact, in our old work, Paran's work and work from others, we really didn't see much falciparum activity in young rings. It's just there's enough. There's a process that's relatively modest. Ring stage parasites are not very hungry. They're relatively quiescent parasites. So it seems that they manage okay with somewhat decreased hemoglobin uptake. It's in the trophozoite stage, the parasite is taking up very large amounts of hemoglobin. Falciparum activity is dramatically increased. Those parasites, the K13 mutations don't have too much impact on those parasites. It's this subtle ring stage abnormality that leads to the phenotype. And apparently the rings can survive this okay.
Okay, so Phil, I have a few questions if we still have 2 minutes to discuss.
Yeah, I cannot refuse you, Pranav.
Okay. So one is, so how variable this artemisinin resistant across Africa if you put together data coming from different groups?
Yeah, good question. I mean, this is all in East Africa right now. The data are spotty and we need more surveillance across Africa. But right now, as of today, my knowledge is Rwanda, Uganda, and Eritrea/Ethiopia. a little bit of spread to parts of Tanzania that are right next to Rwanda, but that's it. These are very circumscribed, and we don't know why. I gave you my hypothesis that we think in Uganda, malaria epidemics helped lead to selection, but of course that's just a hypothesis. And why haven't we seen this in other parts of Africa? I don't know. We may soon. In fact, I would guess we will, and maybe it's already brewing in some other places, but I've certainly seen recent data from various parts of Africa without changes in K13. So it's primarily East Africa right now.
Okay. So,
which has which I should say has made our life very interesting working in Uganda.
Okay. So I have a follow-up question, and I think you talked about it in few slides, that whether your panel of 50 genes includes the genes of ubiquitin-proteasome system, autophagy, and protease pathways, that it is possible that it seems the most of the group maybe are focusing on this K13 mutations, but then the resistance could be there, may not be as much as confirmed by K13 by mutations in other genes.
Yeah, well, our panel, good question. Our panel was carefully thought out. I will add, our panel has another role, which is to allow us to look for mutations that have impact on new MMV compounds. So it includes things like PFA-TP4, kinases, other genes that we know are targets for MMV compounds. But nonetheless, we do include multiple potential resistance mediators. Regarding the ubiquitin pathway and other pathways involved in protein degradation that may play a role, they're not fully represented. That's not our main focus, but some of them are. It's a little unclear to me whether those are just downstream effect. Artemisinins clearly, looking at artemisinin resistance, we do see changes in those pathways, but is that a downstream secondary effect, or is that a primary effect on the parasite? We don't know the answer. My impression is that the K13 story is very solid, especially in Southeast Asia. K13 mediates this process. I mean, there's definitely messiness in the data. So you always see some resistant parasites that have a sensitive phenotype or the other way around, but still overall the data are quite solid. So I think K13 having the primary role is very satisfactory, but there clearly are other factors that we don't fully understand. So that kind of study is very important. A problem we've had recently is that, I talked about this with lumefantrine, it's also true with artemisinin, it's hard to get stable phenotypes. Things are not straightforward. So most of you are basic scientists. The world of studying field samples is not so clean. We work a lot with David Fiddick's group in New York, who are really good at transfecting parasites, looking at the effects of mutations on parasite phenotypes. And it's not simple. You take the 580Y mutation and stick it into a parasite, your phenotype will vary depending on which parasite you stick it into. And so the background is really important. And so background is the entire genome of the parasite. We don't know what matters, but we know that sometimes you take a mutation that we know mediates resistance, but in certain backgrounds, the parasites are not resistant. So we still have a lot to learn. There's clearly a balance between resistance and fitness, but it's even more complicated than that. Different backgrounds dramatically affect the phenotypes. It makes it very hard to study this stuff. You can't just say, aha, this is the mutation, stick it into 3D7 and see what happens. Sometimes that helps you, sometimes it doesn't.
Okay, so I have last two questions for me. One is there any association of ACT partner drug on the different mutations you are getting on in CALS13 or the mutations? Because it seems there are different ACT combinations being used for treatment in different parts of the world.
Well, in Africa, it's mostly artemether lumefantrine. And what I showed you here is looking for associations between high lumefantrine IC50s and mutations in the panel that we're sequencing. And there were a lot of interesting proteins here that had modest associations like MRP1, MRP2. You see even the falcipanes down at the bottom, but those are pretty modest associations for this kind of analysis. But you see for K13, it was 6 times 10 to the minus 8, a very strong association between 469Y mutation and elevated IC50 for lumefantrin. Even though, again, I wouldn't call this a lumefantrin resistant parasite. It's just a less susceptible parasite. So we see an association with K13 and lumefantrin, but as I mentioned before, I kind of suspect that's not, oops, I think I'll stop sharing here. Should have done that a while ago. That's not,
So Phil, you need to unmute. You got muted.
somehow I muted myself.
Yeah.
Probably that's not causal. Probably it's just an association, but we don't really know. There's a lot we don't know here. There's a lot of interesting work for basic scientists here, obviously.
So the last question is from Deepak. So his question is, does one observe convergence of different mutations towards multi-drug resistance in a specific geographic area?
Say that again, please. I didn't quite
So
follow
his
it.
question is whether there is convergence of different mutations towards emergence of multi-drug resistance in a particular area.
Well, in Southeast Asia is our laboratory. That's where we see things first. And in Southeast Asia, they had artemisinin, what we now call partial resistance, for about 10 years. In Cambodia, they switched to DHA piperacin as their regimen. And within a couple of years, they started seeing piperacin resistance, not super high level resistance, but IC50 is going from 5 or 10 nanomolar up to 30 or 40 nanomolar. And then they saw the ACT failing completely. They saw greater than 50% of patients failing therapy with DHA piperacin. So it really does appear that the real recipe for trouble is when you have artemisinin partial resistance and then get resistance to a partner drug. These drugs are not well matched pharmacologically. Artemisinins only last in your bloodstream for a few hours. The partner drugs last for a long time. So the partner drugs are definitely at risk of resistance selection. And when you get that with the underlying partial resistance to artemisinins, then drugs actually fail. We don't think we've seen that yet in Africa. If we do, it will be a disaster because, again, 95% of malaria is in Africa. So that's what we're most worried about right now.
So last question from me, it's not really a question. So do you think if iron supplementation as part of ACT can help in reducing artemisinin resistance? I don't know if it is being done.
I have no idea. I mean, as you know, with a number of infectious diseases, iron is sort of a two-edged sword. Iron can help you or it can hurt you. There's some infections that get worse when you do iron supplementation. And that may be the case with malaria. There's I think a complicated, controversial literature on iron and malaria. I'm not really an expert. But my answer to your question is, I don't know. That's a good.
So it seems we don't have any question and we are also running out of time. So Saujanya.
Yeah, Puran. I
Yeah.
think we can wrap it up. We have no more questions, neither on YouTube or on Zoom. I would like to thank both the speaker, Dr. Philip Rosenthal and Dr. Puran Singh Sajwali, for being here with us today. Thank you so much.
Thank you all. It's been a pleasure. Bye-bye.
Thank you, Phil. Hope to see you soon.